I have been working in EPR since my PhD. It was this technique really attracted my attention because it allowed me to have a really important tool to be able to access microcosm and the protein systems I have been working on. For me, it was always interesting because I could use biophysical tools and spectroscopic tools in order to be able to understand important biological questions. A very interesting aspect of the EPR methodology, which is quite unique, is that it allows us to understand the dynamics of complex proteins. It's very important, apart from having static images of proteins, to be able to understand their motions, how they move, and therefore understand their function. So EPR allows us to monitor conformational ensembles of conformations of these proteins and also their oligomeric state, which is quite important for assembly of these complexes. This is something quite unique since it gives the opportunity to the scientist to detect signal only from the spin centers that we engineer within these complex proteins, and this allows us to be able to detect EPR signal from these particular regions. EPR was instrumental really, and allowed us to measure distances for the first time within a multimeric system, in particular the mechanosensitive channel of small and the mechanosensitive channel of large conductance we're investigating in our lab, and this allowed us to see the change in conformation upon tension application. For that you need really high resolution, and EPR allowed us to have this angstrom resolution and detect minute helical movements of these very important systems. The vast majority of the systems we're working on are drug targets. Therefore, we can design more effective pharmaceuticals and also make the existing, for instance, antibiotics more effective. At the moment, we're working mostly on mechanosensitive ion channels. In the future, we would like to have two novel directions within my group. One would be to study other membrane protein classes, such as transporters or receptors. These systems are also very important drug targets for various diseases, such as diabetes or cancer. And the second direction we would like to really pursue would be to be able to do this kind of EPR measurements in cellular native conditions. At the moment, in order to be able to perform these experiments, we have to isolate these proteins from a cellular environment. In the future, we would like to be able to do the same kind of measurements within real intact cells. The new instrument has a 300-watt amplifier, which increases the sensitivity, and this is quite key for being able to detect signal from lowly expressed membrane proteins that we have been working on. Therefore, we expect that now we will detect signal from these systems, whereas before we couldn't. And that will allow us to enter a completely new world of protein science. This new center, BioEmpire, which I lead, it will be devoted to challenging biological applications. So we're really focusing on that, and then we are going to have interactions with other pulsed EPR centers within the UK, such as St Andrews and Imperial College as well, in addition to Manchester National facilities. And this is key for the community as well, and new talent, and we have to train the new generation of students and postdoctoral researchers to enable progress in the field.